Hey everyone. Yes, I'm Holly and I'm from James Cook University on Gimoy country, Cairns, and I'd like to start by acknowledging the traditional owners of the land on which I stand. So I'll be talking about my PhD research, investigating the effects of insecticide exposure, pathogens and parasites on bee diversity, abundance and health. So when you think about bees, probably the honeybee pops to your mind, but in Australia, we actually have over 1,800 species of bees, and in the world, we have over 20,000 species. And most of these are actually solitary, which means they don't live in a colony. Usually it's a female bee, and she'll provide nectar and pollen for her babies, and she'll build a little nest for them. And so these bees are very diverse in size, form, colour, and behaviour. And this is just a few of the beautiful bees that we have in Cairns. But unfortunately, bees are under threat, and this is due to multiple stresses, including insecticide exposure, pathogens and parasites, poor nutrition, climate change, introduced species, and loss of habitat. And so my research will focus on insecticide exposure and pathogens and parasites. So both of these stresses have sublethal and lethal effects on bees, but limited studies have looked at the combined effects on non-apis bees. And a non-apis bee is any bee that's not a honeybee. So bees are exposed to multiple stresses throughout their life, and the combined effects of these stresses could be synergistic, which means they could add on top of each other and make the effects worse. So the first aim of my research is investigating whether insecticide exposure is associated with decreased bee diversity, abundance, and functional diversity. And to do this, I'll be conducting a field study in two contexts. The first will be rainforest treated with insecticide for control of invasive species. And the second will be rainforest adjacent to sugarcane treated with insecticide for control of pests. So I'll use complementary sampling methods, including malaise traps, vane traps, and trap nests, and my field study will run for a year. So my second aim is investigating whether insecticide exposure is associated with decreased bee predator abundance and pathogen and parasite prevalence. And so the insecticides used in these contexts are non-discriminatory. So they could affect any organism in the ecosystem. And this could include bee predators like wasps or kleptoparasitic bees, also possibly skinks or ants or mites as well. And then my third aim is investigating the effect of nesting density on bee predator abundance and pathogen and parasite prevalence. And so you might have heard of bee hotels or insect hotels or pollinator B&Bs. They come in lots of different shapes and sizes, commonly sold at Bunnings or online. So these hotels utilize some bees' nesting preferences, and they're often claimed to be good for bee conservation. But they artificially aggregate nesting sites above densities naturally available for cavity nesting bees. And this could possibly increase pathogens, parasitism, and predation. So I'll be conducting an experiment in the field to test this, and I'll be using two designs of bee hotel. I'll have an aggregated design, which will be 12 blocks clumped together. I know that's only 9 in the picture, but I will use 12. And then a dispersed design, which will be the same type of blocks, but they'll be dispersed across a grid, each 5 meters apart and attached to individual trees. And these are some of the design features, and they'll be checked every 3 months. So once a bee nest is capped off in the field with resin or leaves, I'll be able to tell that it's a bee. I'll remove the straw that will be inserted inside the hotel and take it back to the lab, and I'll rear it until an adult emerges, and I'll also dissect the nest contents to extract DNA and RNA. And then I'll test for fungal parasites, bacterial pathogens, and common Apis mellifera viruses. And this part of my project was very generously funded by the Australian Wildlife Society. So thank you so much for that. And on the right, we have some of the pathogens that I'll be looking for. So far, I've been very busy building hundreds of bee hotel blocks, and I've also piloted my methods for checking, and I've had a few bees visit. This is a fire-tailed resin bee in this picture here. And then I've also sampled some bees in my vane traps, and I'll be starting my field study in the next few months. So stay tuned to see what I find. Thank you.